friends, today we are discussing about the sources of drugs, organized drugs and unorganized drugs. So, if we talking about the sources of drugs, there are mainly many sources of drugs from the pharmacological point of view. But if we are talking from the pharmacognostical point of view, we have mainly five main sources of drugs. So, these are plant sources, animal sources, mineral sources, marine sources, and plant tissue culture sources. So, if we start with the plant source is one of the major source. I would say 80 to 85% of the drug comes from plant sources. They are generally useful for various type of diseases. If we look at our nature, it always exists in a balance. You know that the animal needs oxygen for breathing and they breathe out carbon dioxide, and plant breathe carbon dioxide and they breathe out oxygen. So in this simple example we can understand we are dependent on each other. So same way all the element or illness we have treatment from plant sources. If we look at the traditional system of medicine like Ayurveda, Siddha, homeopathy and Unani systems are based majorly on plant drugs. Majority of secondary metabolites are found in plants like alkaloids, glycosides, flavonoids, enzymes, volatile oils, etc. So here I taken some examples for understanding of plant sources drugs. We can categorize drugs based on their plant part. If it is leaves, then the example is digitalis, eucalyptus, tobacco, and the chemical constituents are digitoxin, digoxin, eucalyptol, nicotine. Then, if it is flowers, then the example is vinca rosea, and chemical constituents is vincristine, vinblastine. If the plant part is fruits, then the example is Physostigma venenosum, then the chemical constituent is Physostigma. If the plant part is seeds, then the example is Nux vomica, chemical constituent is strychnine. If the plant part is bark, then the example is Cinchona, Atropa belladonna, chemical constituents are quinine, atropine. If the plant part is stem, then the example is Chondrodendron tomentum and chemical constituents are tubocurarine. Second category is the animal sources. So in animal, also either the animal product or certain animal parts, they can be used as the drugs. Generally, they can be animals' body secretions, fluids or glands. For example, for animal drugs are insulin, heparin, adrenaline, thyroxine, cod liver oil, musk, beeswax, enzymes, and antitoxin sera are some examples of drugs which obtained from animal sources. Like plant products, drugs from animal sources may be crude, that is unrefined, or refined material. Then the third category is the microbial sources. So penicillin produced by Penicillium chrysogenum, streptomycin from Streptomyces griseus, chloramphenicol from Streptomyces venezuelae, neomycin from Streptomyces fraderi, bacteriocin from Bacillus subtilis, xanthan polysaccharide gum secreted by Xanthomas campestris, dextran, which is polysaccharide of glucose synthesized by lactic acid bacteria, leuconostoc mesenterides, Streptococcus mutus, and Lactobacillus brevis, etc. Then the fourth category is marine sources. Marine sources means the sources from basically present under sea and ocean. So flora and fauna have many plants as well as animals which produce bioactive compounds. Floral, sponges, fish and marine microorganisms produce biologically potent chemical with interesting anti-inflammatory, antiviral, and anti-cancer activity. For example, curacin A from marine cyanobacterium Linkbia majuscula, eleutherobin from coral Eleutherobia sp, diasporomerolite from marine sponge Riscoderma resoluta, so potent anti-tumor activity. When we have the mineral sources, minerals means both metallic and non-metallic minerals have been used as drugs since ancient times. Our body requires trace elements of minerals in order to maintain homeostasis. Examples are ferrous sulfate in iron deficiency anemia, magnesium sulfate as purgative, magnesium trisilicate, aluminum hydroxide, and sodium bicarbonate as antacids for hyperacidity and peptic ulcers. Then we have the synthetic and chemical derivative. A synthetic drug is produced using chemical synthesis, which rearranges chemical derivatives to form a new compound. One of the earliest synthetic drugs was sulfonamide, which began with the synthesis of frontosyl dye. Other examples include acetyl salicylic acid, which is aspirin, oral antidiabetics, antihistaminics, thiazide diuretics, chloroquine, chlorpromazine, general or local anesthetic, paracetamol, phenytoin, etc. Then is the semi-synthetic sources. Semi-synthetic drugs are a hybrid and are generally made by chemically modifying substances that are available from natural sources to improve its potency, efficacy, or reduce side effects. In semi-synthetic drugs, the nucleus of drugs obtained from natural source is kept intact, but the chemical structure is altered. Bean is the semi-synthetic sources. Semi-synthetic drugs are a hybrid and are generally made by chemically modifying substances that are available from natural sources to improve the potency, efficacy, or reduce the side effects. In semi-synthetic drugs, the nucleus of drug obtained from natural source is kept intact, but the chemical structure is altered. Examples of semi-synthetic medicine include heroin from morphine, bromoscopolamine from scopolamine, homatropine from atropine, ampicillin from penicillin, etc. Now we are discussing about the organized and unorganized drugs. If we look at the food drugs, it can be divided into organized drugs, which is cellular drugs, and unorganized drugs, which is acellular drugs. Now we are discussing about the organized drugs. Organized drugs consist of the cellular organization in the form of anatomical features, mostly the crude drugs from the plant sources. Cinchona bark, sandalwood, senna, digitalis leaves, Nux vomica seeds, and Rauwolfia roots. Microscopical anatomical studies are preeminent for such crude drugs. This can be used directly in medicine or can be used by modifying or by extracting the active ingredient from it. Then the unorganized drugs. Unorganized drugs do not have the morphological or anatomical organization as such. These are the products which come directly in the market, but their ultimate source remains the plant, animal, or minerals. Microscopical studies are not required for such food drugs. These include products like plant exudates as gums, oleogum resin, plant lettuce, like that of opium, aloetic juices like aloes or dried extract of black and pale catechu, agar, alginic acid, etc. products coming under this group.